we'll get started. Okay, so I'm really interested in really working out the fine level of analysis of the circuits and the details. I'm interested in the molecular pharmacology. I really want to understand how the brain is wired. There are tremendously powerful tools to study this in mice. We can do these with genetic approaches. We can do these with viral approaches. We can use optogenetic approaches. And elements of this are likely to be interesting to many of you. And we can talk about that at lunch or whatever you want to do. Understanding complex emotional experiences by using animal models obviously has its limits. We can get a lot of insight, a lot of information, but ultimately we're going to need to get into people if we're going to want to ask really interesting questions. You can't get to the same level of circuitry when you do studies in humans, but we're developing tools, the field is developing tools to try to ask questions about what does it mean for depression? What is the role of dynorphin in mood disorder? What are the other stress hormones and their role? How are the neural circuits altered in individuals who are depressed so that dynorphin might be functioning differently? We know that individuals with depression are really hypersensitive to stress. They react badly to adverse circumstances in their lives in an exaggerated mode. But our insights about the nature of depression are just as crude as our insights about the nature of addiction, and what there are tremendous opportunities for the next generation of scientists to really use these evolving tools to answer very, very difficult but really, really interesting questions. So before I make the next part of the talk, which what I want to do is leave the circuit analysis and move on to how can we develop these insights into therapeutically valuable tools. Obviously, if kappa receptors are activated by stress-induced dynorphin, and it has all sorts of effects on reward valence and the risk of drug addiction, could we develop new therapeutics to treat drug addiction by blocking the kappa receptors? That's what I'm going to talk about next. Before I get to that, though, I wanted to kind of open it up and hear something from you based on the kind of superficial analysis that I just presented. Are there any topics that you think are worth a little bit more discussion, or that you're interested in trying to understand in your own research that you want to bring out for the group?
Charlie, there was a question here about self-administration and kappa knockouts.
Okay.
So maybe
What would you predict?
Where if they decrease the balance of the cocaine, but it's in stress-dependent manner. I think they won't differ in basal conditions, they might differ in stress conditions.
Okay. How would you test it?
But I would need a paradigm in which the mice actually would need to set up under stress conditions to see the increase, so kind of not really the reinstatement, but well, I'll go first for stress-induced reinstatement, but that's not maybe the best. But that would be my first pass.
Sure.
Stress-induced reinstatement.
And those experiments have been really informative. If you look at just cocaine CPP or heroin CPP or alcohol CPP, kappa knockout doesn't make any difference. The rewarding valence of these drugs isn't affected by the absence of the kappa receptor. But if you stress the animal, the stress-induced potentiation of nicotine reward, stress-induced potentiation of cocaine or alcohol reward, all of that is missing in a kappa knockout animal. If you give an animal access to leverpress for cocaine, they like it just fine. They acquire the cocaine self-administration behaviors the way a normal animal would. They like cocaine just fine. And in a standard short-axis paradigm, you couldn't tell the difference between an animal that had kappa receptors than one that didn't. It's only when you get into extended access, and exactly what's happening in an extended access is not very clear, but one of the things that seems to happen is that they get to much higher levels of drug, or if you deliberately stress the animal, then you can begin to see changes in their behavior. I showed you one bit where they actually showed a different break point if they had been stressed. So they experienced the cocaine, they like the cocaine, but they're willing to work a lot harder for the cocaine if they have a stress experience, and that's missing in a kappa knockout animal. So if you were to imagine taking a kappa antagonist as a human, it's not as if you're going to be more likely to abuse drugs, but that you are less likely to abuse drugs if your motivation is because of your dysphoric state or your anxious state. And it's more likely that the withdrawal reaction that you get in a dependent state may be attenuated in ways that might make it less likely for you to reinstate drug-seeking behavior. We can extinguish self-administration behaviors, and then we can stress the animal and they'll reinstate. That's been shown for cocaine, for alcohol. I don't know. We talked about whether it's been done for nicotine. It's not yet been done for nicotine. We predict that it would be important for nicotine, but that's not yet been shown. So stress-induced reinstatement, I think, would be a very important issue for a kappa antagonist. But I think that that's getting to the motivational aspects. Any other? Yeah.
That's a very nice question. So, has Norby-NI been given to humans?
Unfortunately, Norby-NI is not a very good, it's not been approved for use for humans. I don't know that the safety and has been established. It's not a complicated compound. It's essentially, Phil Portuguese took naltrexone, which is given to people, and he made a naltrexone dimer. So there's no reason why it shouldn't be safe for people, because naltrexone is safe for people, but it's never been done. And the Food and Drug Administration is pretty fussy about how you actually demonstrate safety before you're allowed to do a clinical study. There are kappa antagonists that are in development, and I'm really impatient to know what they're doing. But I don't do that kind of work, and so I have to sit and be patient. But it would be really important. We were talking about this last night. Really important to get drugs into different trials and see what they do. The predictions from the preclinical data are very clear. That you will have stress-induced mood disorders in people that might be very sensitive to the dynorphin component. But exactly how much, we don't know, because we don't know to what extent the human experience is clearly perfectly modeled in our rodent experiments. We're skeptical that they're one-to-one going to match, but we want to know to what extent they have validity. We won't know until we actually get drugs that the Food and Drug Administration approves into people. Other questions? Sure.
I might be mistaken, but I wonder if you might give more story into this. I think this is like a quite validated hypothesis that dynorphin system has been thought to counteract the over-activation of dopamine system, and that what it does is a negative feedback to an decrease of dopamine, increase of comments. So it seems that you haven't talked about it much today. Is it like kind of abandoned field, or is it not anymore?
Well, the hypothesis was an inference based on evidence that when you activate the brain with psychostimulants repeatedly, there's an increase in dynorphin message. And then The second piece of evidence is that if you give a kappa agonist pharmacologic administration at the same time you give a psychostimulant, you have a reduced rewarding effect of the psychostimulant. And so the idea that there was an opponent process was supported by those two lines of data. But there was no direct demonstration that when the body uses dynorphin as a neurotransmitter, that its role is to damp down the cocaine reward or the amphetamine reward. And so that was what we tested. And what we found instead was that actually when you're in a dynorphin elevated level, you're hyper responsive to the rewarding effects of drugs like cocaine. And so the rewarding valence of drugs like alcohol and cocaine and heroin are actually increased. So rather than it being an opponent process, the agonist, the dynorphin endogenous system, is actually priming the system, inducing what I believe is craving and inducing what I believe is a likelihood of relapse and reinstatement, to the point where dynorphin is not acting as an opponent process, but instead acting as a motivator. When you talk about that, then you'd want to be able to say if that model is true, then a kappa antagonist given to a person who's stressed would reduce their risk of drug abuse, would reduce their risk of self-administration or relapse. Whereas the other idea was that what you'd want to be able to do is give an agonist, not an antagonist. And so the question will be resolved when we know which is better for drug addiction, treatment of drug addiction, an agonist or an antagonist. The data that I presented all says that it's an antagonist that would be better, but there are others who are still willing to try an agonist for the treatment of drug addiction because they interpret the preclinical data in a different way than I do. So it's not resolved and won't be resolved until we do the studies in people.
So, yeah, there's been a few limited trials of kappa antagonists and things that have been done, certainly into different populations. But there is the functional kappa antagonist, where there's a combination of naltrexone and buprenorphine.
Absolutely.
Do you have an opinion?
Love it. So buprenorphine is a great medication that's an alternative to methadone. It's a complicated drug because not only is it a very strong kappa antagonist, it's also a weak mu partial agonist and an ORL partial agonist. And so interpreting why buprenorphine does what it does to people is not very clear. But what is clear is that buprenorphine reduces people's desire for opiates. And there have been some interesting studies, really open label, not carefully double-blind controlled, where the mu partial agonist effects have been occluded with the addition of a mu antagonist. So now you've got a combination of a drug that's got a mu antagonist and a kappa antagonist. And for those individuals who take that preparation, there are reports of reduced cocaine craving and reduced cocaine relapse. And I would predict that that would suggest that a selective kappa antagonist would have the same effects, but cleaner. There are some really interesting open label trials saying that treatment-resistant depression, patients who are depressed, but the standard methods of SSRIs or reuptake inhibitors don't really work for them. When they're given buprenorphine with the mu receptors masked, they also show antidepressant effects for that. And so I think that there's a little bit of preclinical data and early clinical trial data supporting the concept that a kappa antagonist could be effective in both depression and in treatment of addiction. But as we've just talked about, we need to get drugs approved by the FDA and into as many clinical trials as we can. I mean, I was talking last night, I mean, the first clinical trial that I would love to see happen is that if an individual was heavily nicotine dependent, and if you look at what happens with a nicotine dependent person, when they are in between bouts of their drug, they show a period of anxiety where they're craving the nicotine. But if they want to quit the nicotine, it's hard. They just relapse because the compulsion and the cravings are so strong, and they're triggered by the adverse effects of withdrawal. I think that that would be very interesting based on what we know dynorphin contributes to those feelings to try a kappa antagonist in that population. Same thing would be true for cocaine or heroin as well. Yeah.
So for these preclinical models of depression, so thinking about how the antagonist might work, does it prevent the acquisition, do you think? Or do you think it can reverse, like once you've induced like a social defeat, would it then reverse like this before and after effect?
There's not a lot of data that talks about after the individual is depressed, what happens, can you reverse the actions? There's some indication that it can work. Obviously, if you give the drug beforehand, you can help. So I would just be speculating to try to say if you've got somebody who already has a high dynorphin tone, to what extent would the kappa antagonist be useful? We don't know that because the animal models that we've got pre-treat with the kappa antagonist. But I would predict that for an individual who is depression vulnerable, they're getting stress-induced dynorphin release as they go through their day, and it's contributing to their mood in ways that might be ameliorated by an antagonist. But we won't know until we get these drugs into people. Yeah.
I have a question about the mechanism. In the last slide, you showed that you believe that there is an increase in the serotonin reuptake.
Yes.
Why not a decrease in the release? It's the most common mechanism for EI protein effect.
Well, we know that the dynorphin will act presynaptically to reduce release, and I thought initially that that would be enough. But when you look at the role of P38, it turns out that P38 activation is required. There's no evidence that P38 is involved in the inhibition of serotonin release. And so that is probably an ion channel effect directly on the release and fusion of the serotonergic vesicles. We haven't proved that yet, and that's one of the things that we're doing in the laboratory right now, is to try to look at the differences between dynorphin effects on release mechanisms and dynorphin effects through P38 on reuptake mechanisms. And answering that question experimentally is what we'll be doing in the next 6 months.
What is the idea? Because I mean, how you can design an experiment?
How will we design the experiment? Well, what we're using is optoactivation of the serotonergic pathway to actually stimulate release. We can measure it with fast scan voltammetry and carbon fiber recordings to measure the serotonin in the intact animal in vivo as they're walking through their cage, and when you stress them, to what extent is serotonin release changed, and when we activate dynorphin release optically, can we influence the serotonin reuptake, and can we correlate the ability of dynorphin to activate kappa receptors on those nerve terminals to suppress serotonin release with the behavioral manifestations that we see. So we can manipulate very selectively elements of the circuit now with the modern tools that have recently been developed. And we'll know the answer exactly to that question within the next 6 months. Okay.
Sorry, is P38 required for kappa internalization?
No, that's an arrestin-dependent mechanism. So if you block P38, it doesn't seem to influence the ability of the kappa receptors agonist-stimulated internalization.
Okay, so it doesn't mute, but
For the agonist, what happens is that the arrestin is already bound, and it is involved in, so that's a GRK-dependent event. Okay, any other questions or comments? Okay, so I've told you just briefly in a fairly superficial way some of the data that says that dynorphin is involved in anxiety behaviors, depression-like behaviors, potentiation of addictive drug-seeking behaviors for nicotine, cocaine, and ethanol, reinstatement from extinguished drug-seeking, that's cocaine primarily, but I think also ethanol as well. All of these effects are blocked by kappa antagonists, and that says that a kappa antagonist might have potential therapeutic utility in these disorders. We talked earlier in the morning about the comorbidity between depression and addiction, anxiety disorders and addiction. And so clearly if we had a way to manipulate both the addiction risk and the mood disorders, we might be able to help individuals in ways that we can't currently do. And so the idea then is, well, how do you make a kappa antagonist, and how do these kappa antagonists work? Kappa antagonists have been around for about 20 years. The first kappa antagonist that was developed, selective kappa antagonist, is Norbi. But as I told you, they've got this really amazing property that a single injection of a drug can last over 3 weeks. In some primate models, it can last many months. So the idea that these drugs, after a single injection, have very long durations of action have been a puzzle. When we first presented the data about 10 years ago at a scientific conference where there were representatives from NIDA, they said the Food and Drug Administration is not going to let us put these long-acting drugs into people unless we can explain why it is. They're not drug-like in a standard way, where if you've got a typical drug, the drug effect lasts a little while, and that's important because if you've got adverse effects for the human, you want to be able to take them off the drug before the adverse effects really become overwhelming. And you can't with a drug that lasts more than 3 weeks. If they develop an adverse reaction, you're really stuck. So we needed to explain how that might be. What we found in a series of studies was that the kappa antagonists are more than just competitive antagonists. Remember from your pharmacology class that when you have a mu-opioid receptor and you put naloxone on first or an antagonist on first, it works because it occupies the binding site and morphine can't bind and morphine can't activate the receptor. But what happens with Norbi turns out that when it binds to the receptor, it isn't just simply turning off competitively inhibiting the access of the agonist. It actually, in a ligand signaling, directed signaling way, will activate c-Jun kinase, and c-Jun kinase will inactivate the signaling system in a way that results in receptor inactivation. And so long after the Norbi comes away from the receptor, the receptors are inactivated. That's really pretty surprising. It took us a lot of work to actually develop this idea, but the concept of functional selectivity is really important, and molecular pharmacology and drug development is now driven by the idea that when you activate the receptor with drugs and you look at the signaling consequences, they're not all equivalent. It used to be that we would think that agonists differed solely in terms of their efficacy. Some drugs strongly activated the receptor and they'd be full agonists. Some drugs were weaker efficacy, they were partial agonists, they would produce a smaller response. Some drugs were antagonists, and some drugs were inverse agonists. It was all just fairly linear in its dimension. But now that we know that you can activate different signaling pathways following the activation of one receptor, at each individual receptor you can have 5 different effects, and an individual drug could activate with different efficiencies each of those different circuits. And what Norbi does is it activates the c-Jun kinase pathway circuit without activating the G-beta gamma circuit in a way that makes it a functionally selective drug. The kappa antagonists that have been developed include Norbi and JD-TIC. JD-TIC was in clinical trials a year ago, and unfortunately there were cardiovascular effects that appeared when people were taking these drugs, and the drug was abandoned. There's other dimensions of these drugs that we're hoping to be able to exploit and try to get around that.
Were these cardiovascular effects dependent on the kappa receptor, or it was just...
It's not at all clear. In none of the preclinical models, and none of the mice or primate models, were there any evidence of cardiovascular effects. And it's not at all clear that it was even a drug-specific event. It could have been just a coincidence that it was just a small number of patients, and 2 of the patients with the drug showed these things, but in that particular patient population, it could have been that those were just part of their normal variation. Or it could be that there really is a kappa-dependent effect on the cardiovascular system that we've never appreciated because those particular individuals had some other cardiovascular pathology or issues that made them more vulnerable to that. The study was stopped, and it's unfortunate, but that's...
Pre-tested in space. Anyway, there's a lot of upset about this.
What was the population they cohort that was used for this system?
I don't remember all the details in the population. They were described in the...
Was it phase one? I mean, is it normal individuals?
They were normal individuals. As far as we could tell, they were normal individuals. I don't know that they weren't youthful individuals. They were middle-aged individuals. And so the variants, who knows what they were? They were volunteers. And so they're not randomly selected. They're people who actually agreed to participate in this study. They were healthy in every other way as far as the screening could show, but obviously it was difficult.
With very low doses.
And the cardiovascular effect was QT prolongation or?
No, it was an series of extrasystoles. So they're extra beats. It's not implausible that there are effects of kappa on the heart. There's dynorphin in the heart, and there are the possibility that there could be changes in excitability in the heart. The drug didn't show any of those effects in rodents or in primates, but human heart may be different. Okay.
Which would be difficult for any kappa antagonist. Nothing.
That doesn't produce major cardiovascular effect.
Right. Right. Because it's a really novel idea that kappa antagonists actually have this property, I wanted to just take you through a little bit of the data to support the explanation. When we first saw this observation, it was really quite a puzzle. I mean, let's back up and say you inject the drug, and it lasts 3 weeks. That's really weird. We had no idea how that might be, and when I talked to the medicinal chemists about this idea, the first thing that they said is, well, the drug is just slowly cleared. Some drugs remain in the body for a long period of time. That must be what's happening here. And that's a pharmacokinetic explanation. And it's hard to really argue with that explanation because you can't really tell how much drug is present in the site of action. It could be dissolved in the lipid bilayer. It could be trapped in the brain. It could be at that concentration that's effective but too low to measure chemically. So that was the explanation that people believed who hadn't really explored it fully. The second is that in vitro, in HEK cells or in transfected cells, the drug reverses relatively quickly. It doesn't covalently bind to the receptor, and it can be washed out of our tissue preparation. So maybe when you give it to a whole animal, or what's happening is it's being metabolically converted into something else that is long-acting. And the third possibility is that it's actually not very simple, and it is really a pharmacodynamic, and that the drug can actually do something even though it's not an agonist, doesn't produce analgesia. It does this ligand-directed signaling. The field has absorbed this idea of functional selectivity only reluctantly and only recently, and so during the 20 years that we've known about this compound, it's only been during the last 5 or 6 that the concept of functional selectivity is now the new dogma. But in the old dogma, it just wasn't acceptable. But in 2007, when we were asking this question, these were the 3 mechanisms that we tried to resolve. The answer was that when we were doing a set of studies with transfected cells, and we would look to see how kappa receptor activation causes the activation of different MAP kinase cascades, we can easily demonstrate kappa receptor activation at ERK, and that happens in a GRK and arrest-independent mechanism. And that effect is blocked by norbinide, the kappa antagonist. When we do similar experiments with the activation of P38, kappa agonists like U50 and dynorphin all activate P38 mechanisms in an arrested and GRK-dependent way through a scaffolding process. And so I had an undergraduate student for the summer who wanted to help out, and we thought, okay, what about Jun kinase? And so we were looking at kappa activation of Jun kinase mechanisms, and that was really great. But the control experiment was to block the agonist effect with NorBNI. And what surprised us is NorBNI didn't block it. When we looked at untransfected cells, the kappa agonist didn't activate Jun kinase. And when we did the control experiment with the kappa transfected cells, the NorBNI activated Jun kinase as well. And so around the same time, we were puzzled about why NorBNI was acting in such a long duration way. We had this in vitro data that NorBNI was actually able to activate Jun kinase, and we said, well, maybe the two events are connected. And the answer is that when we block Jun kinase in vivo with a pharmacologic inhibitor of Jun kinase, SP6000, it can make NorBNI into a short-acting drug. Drugs like SP6 inhibitors, as you know, these kinase inhibitors, they're not terribly selective, and they can block all sorts of other cascades. So when you use a pharmacologic inhibitor, you really want to be very skeptical that the mechanism is attributed to just what it says on the label, Jun kinase inhibitor. So we got a hold of the JNK1 knockout animals, the JNK2 knockout animals, and the JNK3 knockout animals. And the data that I'm showing you behind me is that when we looked to see whether NorBNI was a long-acting drug in those knockout animals, it was a short-acting drug in the JNK1 knockout animals, long-acting in JNK2 and JNK3. So that's consistent with the idea that NorBNI activates Jun kinase 1 in animals to produce a long duration of effect. And you can see the ability of that not only in an analgesic assay, but also when you isolate the membranes from the spinal cord after treatment with NorBNI, you can see from the GTP-gamma-S data that the receptor is inactive and that no longer, even in vitro, when the drug is washed out, you can't have agonist activation of GTP-gamma-S. So it's something that's happening with Jun kinase at the receptor signaling event where it no longer functions in the right way. If we treat the animal with a beta-chloranaltrexamine, which is an irreversible alkylating agent which will covalently bind to, with a nitrogen mustard reaction, to all of the opiate receptors, and then challenge the animal with U50 at various time points after chloranaltrexamine, it takes 3 to 4 weeks before the effects are reversed. That's really, really slow. Kappa receptors seem to be turning over very, very slowly. So when you inactivate the receptor with chloranaltrexamine, it takes a long time for the recovery. When you treat the animal with NorBNI, it takes the same long time. And the receptors are not covalently inactivated. If you look at the binding sites, the ability of agonists to bind to the receptors after NorBNI is unaffected. They just don't signal anymore. So we're really interested in trying to understand how the receptors have become inactivated. And the kappa receptor is unique in the sense that it turns over very slowly. We're used to thinking about membrane proteins turning over quite quickly. And in fact, the mu receptor does turn over quite quickly. If you inactivate the mu receptors in the animal, same chloranaltrexamine, within 3 days, the response to morphine is fully back. So different receptors turn over at different rates. The mu receptor turns over really quickly. The kappa receptor, for whatever reason, is synthesized and replaced really very slowly. That's kind of interesting. If you're thinking about, the therapeutic implications, I don't know, I think it might be really an advantageous situation if you could inactivate the kappa receptors. Then it doesn't depend on whether you've remembered to take your pill that morning. You've just got a steady level of receptor inactivation. So therapeutically, you could imagine that a long-lasting drug might be advantageous. But we want to be able to develop short-acting drugs, and now we understand why NorBNI is long-acting. We can select for those compounds that fail to activate Jun kinase. And that data is shown here. So if we look at all the different kappa antagonists that are available, we got them all. In the lab, we got every single one of them. We tried to look at them and figure out which ones would be long-acting and which ones would be short-acting. I don't have the ability, no medicinal chemist was able to look at that and make a good prediction. But what we did was a set of studies where we injected them individually into animals and then asked could we correlate the duration of action, in other words, after the single injection, come back and challenge with the agonist at different time points? How long until the effect of the agonist recovers compared to how much Jun kinase activation could we detect in the spinal cord preparation? And as you can see, there's a really good correlation between the duration of inhibition caused by these antagonists and the ability of the antagonist to activate Jun kinase in the spinal cord. There's no correlation between the duration of antagonist activity and clearance. When we measure clearance directly, it's all over the place. So the pharmacokinetically, the drugs do have different dwell times in the body, but that's not correlated with the receptor inactivation. And so what we now know is that the ligand-directed signaling or functional selectivity is really important for kappa receptor antagonists. And we can think of full and partial agonists, and they have the ability to produce analgesia through a G-beta gamma activated state. We know that high efficacy compounds activate p38 MAP kinase, and that's the aversive quality that I showed you in the first talk, and that long-acting kappa receptor antagonists activate C-Jun N-terminal kinase, causing kappa receptor inactivation. And so based on this, we've got clues as to how to proceed forward in drug development. I'm not a drug company, but I talk to people in drug companies, and you could imagine that you could get a kappa agonist that could be analgesic without being aversive or dysphoric if you can get functional selectivity in a failure of the drug to produce p38 activation. And similarly, you can get either a short or a long-acting kappa antagonist based on the ability to activate C-Jun kinase. Okay, so let me make another point. We can stop and discuss that idea. I think it's pretty straightforward. It was pretty difficult at first to accept the idea that drugs are this complicated, but I think at this stage the world is quite happy to accept that idea. Let me go back and answer this one more question. Go ahead.
For the development of kappa agonists, also peripheral effects are important. I'm talking about diuresis.
Right.
Diuresis
Yes.
was
Oh,
one
yes.
of the main limitations. No?
Oh, absolutely. Absolutely. So Raphael's bringing up a point is that kappa agonists do more than just produce analgesia and dysphoria. They also have effects on the endocrine system. They have effects on the gut. They have effects on cognition. And we are in the process of trying to understand how those different responses are organized at the molecular signaling level. We don't yet know. So whether we could get a kappa agonist that was analgesic and not diuretic, I think is still uncertain because we haven't tested that yet. But what happened was that right after the kappa receptors were distinguished from the mu receptors, when that was discovered in the 70s and early 80s, the pharmaceutical company developed a lot of very selective kappa agonists. They went for specificity, affinity, and efficacy, and all of them were wildly dysphoric, and so it just put an end to drug development. They didn't bother developing a kappa antagonist, whose only role would be to reverse a drug that they weren't going to ever make. So all of that went on hold until recently, and now there's a re-energy where companies are going back and trying to find partial agonists, and hopefully using this ligand-directed signaling strategy, find some that seem to have more selective pathways. Meanwhile, it's up to basic pharmacologists like myself to try to understand something about how the different agonists might produce different responses.
So those new short-acting kappa antagonists do not block the C-Jun kinase?
They don't activate the JNK.
Yeah.
They don't do that. On that list that I showed you was the Lilly, the Lilly compound that you worked on.
And so what pathway do they block?
They're nothing. I mean, they're just a straight competitive antagonist. Just like you were taught 20 years ago in pharmacology class, they are competitive antagonists. And they've got short durations of effect, limited largely from clearance. So just in a very straightforward way. And so as long as, like you find, they have the same ability to block the dysphoric properties of stress, you have to take them more frequently, but when you stop taking them, the drug effect goes away. We don't yet know to what extent Jun kinase activation is good or bad. We haven't tested that yet. Any other questions or comments about kappa receptor and JNK activation. We don't really understand the receptor signaling complex. We know that there's a 7 transmembrane G protein coupled receptor that's important for these signaling events. We know that they bind to G proteins, and when you activate the G proteins, you get the signaling process. We know that they are substrates for the G protein receptor kinase, and when the receptor becomes phosphorylated, arrestin binds. But one of the major advances in our conception about the way the receptor is working are what are the accessory proteins that seem to be important in the localization and in the signaling events and in their regulation. We're learning a lot about how this works. And so understanding how activation of Jun kinase disrupts that signaling complex is going to tell us a lot about the structure of that receptor and how that signaling molecule works in a real neuron.
Charlie, does the agonist activate JUNC as well, right?
It does.
Is it the same JUNC or is it JUNC2?
Same one.
So they both activate JUNC1,
Yes.
but only the antagonist causes the long-lasting.
But the way the agonist activates Jun kinase is in an arrestin-dependent manner.
Right.
So if arrestin is in the way,
It can't be phosphorylating the receptor.
it isn't the same as when Norby and I activate, and arrestin isn't in the way. So we know that we can block U50 activation of Jun kinase because it's through an arrestin-dependent scaffolding process. And when arrestin is in the way, the Jun kinase can't disrupt the signal.
So in arrestin knockouts, you should get long-lasting decrease in.
We should, but we haven't proven that yet.
But I guess G proteins are.
Well, no, if you, in the arrestin knockout, you won't get the U50 to activate JUNC because it needs arrestin in order to do it.
Okay, so the conformations are different.
The conformations are different, and we're busy studying the way that they are different. Okay, new topic. I think the topic relative to an addiction class, I think we've already covered. Kappa antagonists would be really interesting molecules to get into people to see what kinds of effects they have on stress-induced disorders. And when we think about addiction, we know that it's not going to make cocaine or alcohol less fun. It's not going to block addiction because it makes these drugs less rewarding. But when we think about the complexity of addiction and we see these individuals that have progressed to a state where they're no longer finding the drugs fun, but they're medicating to avoid the dysphoria that's a consequence of the changes in the brain, or if they have an underlying depression or anxiety disorder and they're resulting in increased drug addiction risk, then we think that the stress-induced increase in the addictive properties of these drugs might be reduced by a kappa receptor antagonist. But again, that's a theory, and we don't know the answer until we actually do the studies. It could be a lot more complicated. But other issues, I should stop soon. I think I'm ready to stop.
If you're ready to stop, then.
No, I don't want. There are other topics that you guys want to do, and I've used up my 2 hours. But I'll tell you no. But I got more data. No. I will spare you more data, except just to simply say the answer to this question is that other receptors also are regulated by Jun kinase. Once we saw it at the kappa receptor, the next question was, could this happen for any other receptor? The answer was yes. The mu-opioid receptor is also inactivated by Jun kinase. And so when we get back to that list of addiction criteria, tolerance, how does morphine produce tolerance? It does it through this way as well. Okay, I'm going to stop and give you guys a break. We can talk about anything else you want, either in a discussion period now while we're breaking before the next speaker, or we can talk at lunch or on the beach for dinner.
On the beach.
On the beach?
That's a good idea. I think maybe a session on the beach.
Well, thank you so much for your attention. Really.